All right, it is my pleasure today to introduce our speaker and get the slide up here. Dr. Paul Ling. He is an associate professor at the Baylor College of Medicine, researcher and scientist in herpes virus. He does many different things, but a few years ago something happened that got him involved in a different line of his research. There's a problem worldwide having to do with Asian elephants at zoos around the world, including the Houston Zoo. And eventually they discovered that the cause of a particular disease is herpes virus. So who are you going to call? Dr. Ling is the local expert, and he got involved, and he's here to tell you his story. So Dr. Ling?
So today, I'm just going to tell you a story about what we're doing with this project involving the elephants. And if you have questions during the presentation, just raise your hand, shout out to me, whatever, and we'll also have time at the end. And I'm happy to answer any questions you guys have as we go along. So pretty informal. All right. So the title is Saving Baby Elephants from Lethal Virus. I just want to show you a couple things first here. This is what baby elephants are supposed to look like. This is two young baby elephants at the Houston Zoo named Baylor and Tupelo. And at this time they're maybe about a year and a half old. And as you know, elephants love to just play around in water. So what I want to talk about today is I want to go over this story about these elephants. I want to tell you a little bit about what is this virus that we now know is called Elephant Endotheliotropic Herpesvirus, which I know is a mouthful. We just call it EEHV. Why is it significant? Why do we care about it? Why is Baylor College of Medicine involved, or how did I get involved with this? What do we want to do about it? And what have we done to respond to this particular disease that's killing these young baby elephants? I'll tell you a little bit about what we've done, and then also where are we going? All right, so that's the basic scheme of this little talk today. So what is Elephant Endotheliotropic Herpesvirus? And this is just a short little video that kind of describes for you the disease process that's caused by this virus. So see if the video will show. So we think that these viruses are transmitted via trunk-to-trunk contacts between elephants. This virus can transmit through blood vessels in the elephant. It can infect the walls of these blood vessels, this virus can. And as you'll see here in a minute, it can actually cause leakage from these blood vessels, where the blood leaks out of capillaries, veins, and arteries. And this can happen throughout the whole body of the elephant. And as we'll show here in a minute, in particular, all internal organs are affected, but one of the most important ones, of course, is the heart. So if you have these kinds of issues happening in the heart, you get blood leaking from the heart, and these animals actually die from blood leakage from the heart. Now, if you don't mind, actually, I'm going to ask for a little bit of audience participation occasionally through this talk. So can you guys think of any other viruses that may do something like this that we've heard about in the news recently? Any guesses? Ebola? Did I hear Ebola? Yeah. Right. Ebola is one. So this virus looks a lot like other viruses that cause what we call hemorrhagic disease. And hemorrhaging is just a fancy word for saying leakage from blood vessels. Okay? So this virus actually causes a disease that's actually a lot like Ebola. There are other viral hemorrhagic diseases you may have heard in the news about recently. Certainly you've heard of dengue, West Nile virus. These are viruses that can cause these types of diseases as well. This just shows you some pathology from an elephant that died from this virus. You can see these black and very darkened red areas. This is in the large intestine. This just shows you heart. All these black areas are areas of coagulated blood that leaked out from the heart. And you can actually see in other internal organs, this is the kidney, all these patchy areas here, dark areas in these organs are areas of hemorrhage in these particular organs. And what happens is when you get a lot of hemorrhage, the fluid balance within the animal gets all out of whack. And they go into a process, we call it hypovolemic shock, which is a fancy word for just saying fluid balance is all out of whack. And really the animal starts to shut down. All the internal organs shut down when this happens. So actually one of the major treatments for these types of diseases is to maintain fluid support in these animals, either IV, or in the case of elephants, we can actually deliver fluids rectally. Okay, to understand this disease a little bit more, I think we need to actually go back a little bit and think about herpes viruses in general. Like, what do we know about herpes viruses? Actually, we know quite a lot, actually. This is just a picture of a herpes virus, and it shows you that it's got this outer sort of region, we call it a membrane, and there's lots of little things stuck in here we call glycoproteins, which help this virus particle stick to different cells in our body and helps it infect. And inside here, we call this part the nucleocapsid, and the genetic material of the virus is protected inside of here. But essentially, all herpes viruses look the same. They all basically look like this. And the interesting thing about herpes viruses is they're ubiquitous. Well, what does that mean? That means that they're everywhere. All animal species have multiple herpes viruses that naturally infect them. We do too. Most people think, when they think herpes virus, most people think cold sore on your mouth, or people think genital herpes. But that's just two. People are infected with multiple herpes viruses. In fact, it's much more than two. How many people think we're infected with 4 herpes viruses? At least 4. Do you think we're more? How about 5? 6? 7? 8? 8 is actually the right answer. You may not like to hear that. I know you don't want to hear that. But there are actually 8 herpes viruses that infect people. And the thing that you probably don't want to know is that most of us are infected with almost all of them. Let me give you some examples. So chickenpox is a herpes virus. Most of us here have had chickenpox. It can cause shingles. Chickenpox and shingles is actually the same virus. So you get chickenpox, and later on it can manifest itself, process we'll talk about in a minute, we can reactivate and cause shingles. How many of you heard of Epstein-Barr virus? It causes mononucleosis. Maybe some of you have had that, mono. That's actually a herpes virus, okay? Epstein-Barr virus is actually one of the viruses I worked on a long time before I started working on the elephant herpes virus. In addition to causing mono, it also is associated with a number of human cancers. So some herpes viruses cause human cancers. How many of you heard of Hodgkin's lymphoma? 50% of Hodgkin's lymphomas, as part of their cause, is Epstein-Barr virus, which is a herpes virus. Everybody in this room actually is infected with Epstein-Barr virus. It's a different question on why don't we all get cancer from it, but the fact is, again, these viruses are everywhere. The interesting feature about herpes viruses is that they undergo a process called latency. That means you might get some kind of a disease when you get infected with them first, like chickenpox, but then they undergo a process we call latency. This is the ability of the virus to hide in our bodies for a long time, and then it can actually come back later on, a process we call reactivation cause disease. Sometimes reactivation doesn't always cause disease. It can happen at a really low level, and this is how the virus actually spreads from one person to another. And this is spread via secretions, saliva. So Epstein-Barr virus actually used to be called the kissing disease because that's how it's spread. They can be spread through breast milk, saliva, sometimes vaginal secretions. So these are how these viruses are spread. The last thing, which is specific to the elephant herpes virus that I want you to understand, is that, as you all may know, there's actually two different kinds of elephants. There's elephants in Africa. It's actually a completely different species. The Latin name's called Loxodona africana. And then there's Asian elephants, and Asian elephants are called Elephas maximus. They actually can't really even breed between each other. But one of the things we've learned over the last few years is that these elephants carry their own set of herpes viruses. So there are some elephant endotheliotropic herpes viruses, or EHVs, that infect African elephants, and some that infect Asian elephants. But these viruses are different. And the other thing that I want you to remember is that this particular virus seems to be infecting or affecting only Asian elephants, mostly, and it seems to be affecting, in particular, young baby juvenile Asian elephants, particularly between the ages of about 2 and 7 years of age. Okay? So why is it significant? I've just shown you newspaper article clippings here from some baby elephants that died just this year in zoological institutions, either over in Europe or here in North America. Daisy was a 5-year-old female elephant that died, actually it was Mother's Day weekend this year in Albuquerque. There was another elephant at Chester Zoo in the UK, Bally Highway, and then this one was Max, the Whipsnade Zoo over in London. There's actually been 2 more elephant deaths since I made this slide. This was for a talk I did in September. One in the Oklahoma City Zoo, Malie, another 5-year-old female, and then another, this elephant here, which was Bally Highway's brother, Hari Highway. He died just a month ago. So we've had 5 elephant deaths in these institutions just in this year alone, caused by this virus, EEHV. But this virus is actually causing deaths over in the natural range countries for these animals. So again, these are just some newspaper clippings. There's a title here, Herpes virus puts Sumatran elephants under threat. A number of fatal cases have been documented in particular areas of southern India. And then also we're finding cases in Thailand as well. The significance of this disease relative to what we're seeing in captivity is unclear yet, but we just had our first meeting in Singapore 3 weeks ago. I was in Singapore. It was the first Southeast Asian meeting for elephant herpes virus, and we had people from Myanmar, Cambodia, Thailand, Malaysia, Indonesia, wildlife officials, and veterinarians, and they're very concerned about this disease over there. If you look at elephants outside of range countries and just ones in captivity, and you look at their populations, at least in North America, this is just a graph that I want to point out is that the current trend right now of elephant populations outside of range countries, at least in North America, is it's on the decline. And there's a number of reasons for this, but one of these reasons is the impact of EHV. And if you do a calculation and say, well, what if we were able to get rid of EHV as a problem? What would happen? And this is at least one line that we can see. So you can actually see populations would actually start to increase. So EHV is threatening the stability of captive herds. And if we can solve the EHV problem in and of itself, we can actually rescue or help these populations to survive in captivity in places outside of range countries. You might say, well, why do we need to do that? And I'll tell you that Asian elephants are an endangered species. There's maybe 30 to 40,000 of these left in the wild. In some Southeast Asian countries, they're virtually gone. In Vietnam, for example, there's maybe 50 to 100 elephants left at most. Habitat destruction, poaching, all these kinds of things are affecting these elephant populations dramatically, and we may not have elephants in some of these range countries for very much longer. So the work that we're doing, at least for these elephants in captivity, is crucial. Okay, so how did I get involved with working on this? Well, it started actually with, as you recall the video that I showed near the beginning, with Mac, who died in November of 2008. That was the elephant in that video. And the Houston Zoo was trying to establish an active, robust breeding program. They actually had 5 other baby elephants die from EEHV in the preceding 10 or so years. And they were actually at a crossroads, and they said, we need to be more proactive about this. Maybe we need to do something. And one thing led to another, and I'm the local resident expert doing herpes virus research in the medical center, and they approached me and said, can you help us? And I thought about it for a while because I did research on human herpes viruses and human disease. And I was thinking about it and how am I going to get money to do this? I'm at a soft money institution. All my money has to come from grants, all that kind of stuff. And I finally came down to the point, I was at a point in my life where I could take a career risk, and I decided, you know what? Who wouldn't want to help save baby elephants? That's really what it came down to it. And I decided, because of that, I'm going to try to find a way to make it work. I guess I was also at a stage in my life where I was looking for something different. I don't know, some guys my age go buy sports cars or look for a trophy wife or something. And I decided to work on Asian elephants. So my wife was happy about that. All right. So since then, since we started our project, one of the first elephants that was born was this baby elephant here in May of 2010. And the zoo named this elephant Baylor after our program, not to put any pressure on us or anything. Shortly thereafter, Tupelo was born, and early in 2014, Duncan. And you didn't hear it from me. This is top secret. Maybe there's something coming down the road at some point at the zoo in the future. Just to let you know, elephant gestation is 22 months, so that's almost 2 years. So we have a vested interest in keeping these guys alive, okay? All right, so what's my vision? What are we thinking about? How are we thinking about tackling this problem? Or how was I thinking about it? And I actually thought of a research program that actually had 3 phases. The first 2 was to try to develop quick diagnostics to detect this virus and to evaluate some different treatment protocols. And this is what I call short-term goals. I wanted to try to give the veterinarians and the elephant keepers who take care of these animals some tools to help them detect and manage this disease. And these were things that I thought I could do in the short term over a few years, 2 or 3 years, 4 years, the amount of time actually we've been working on this project to help them deal with this disease. And our longer term vision, of course, is to develop a vaccine. What I'm going to tell you a little bit about is some of the things that we've achieved for the short term. Now, as you might know, in human medicine, we have a term that we use for taking things that we do in the lab and applying that to treating that in people. It's called bench to bedside. I've sort of adopted that with a slight modification, and I'm actually calling this bench to barn. Okay, so this bench to barn program that we established has these phases. Okay? So one of the first things that we wanted to do when we started looking at this, if you go back and think about the primer I gave you on herpes virus biology, we kind of wanted to have a look at where can we find these viruses? How might they be spread? And also take a look at what these viruses might be doing, say, in the bloodstream of these animals when they get infected. So we did a couple of things. I guess I'm missing a picture here. We started taking a look at trunk secretions from elephants. And if you've ever spent any time looking at, observing elephants, their trunks are their probe to the world. They're contact with each other continuously with their trunks. That's how they explore their world. And so we actually can instill a little, or pour a little bit of sterile saline into their trunks. The elephants can be trained to hold their trunk up. And then this picture, I don't know why it didn't come out, but they actually can blow it. We actually just take a kitchen bag, and they can blow out that saline in here. You can see the liquid here in the bottom. So they can blow that out. And amazingly enough, we can take that material. It's got dirt and hay and other unmentionables in there. And we can actually process that and extract DNA from that. And we can look for evidence of this elephant herpes virus that's being shed from their trunk secretions. We can also, the elephants can be trained, usually they can give them their favorite food, and they can be trained to stand. This is actually its head, and you can see their ear here. And their ears actually have a lot of blood vessels in the back. The elephants can be trained, they can actually tap into the blood vessels in the back of their ear, and we can take a little bit of blood out of the elephant's ears, okay? So these are the samples that we wanted to take a look at. And we can go back to the lab, and there's some people in my lab can process the blood and some of the trunk washes into DNA or the genetic material where we can look for the virus. And we use a test, it's called qPCR, for quantitative polymerase chain reaction. You don't really need to remember that. There's actually a very, very sensitive test that can pick up small bits, detect small bits of evidence of the elephant herpes virus in different samples. And then you can put it in this very sensitive machine. This is actually me doing this, but that's actually a lie. I don't actually do that. These guys do that. In fact, they don't want me to get close to any of this stuff. They're like, stay away from my stuff. Let me do it right. So anyway, but we put this very sensitive machine that can detect evidence of this virus. So when we started doing this with samples from elephants from the Houston Zoo, what did we find? This is a table. The only thing that I want to point out to you here, this is detection of one of the elephant herpes viruses. There's actually several flavors of them, but this is the most important one, from their trunk secretions. And this is just the frequency by which we could detect evidence of the virus in trunk secretions from most of the healthy adult elephants, okay? So about 40% of the time in some elephants, only 7% in the big adult male bull, Thai, and about 75% of the times from one of the young breeding females, okay? And so what this told us is that many of the healthy adult Asian elephants in the Houston herd probably were carriers. They had been infected with this virus before and were healthy carriers and could occasionally shed them in their trunk secretions. And this is actually maybe what we might expect to find, okay? Because we all do this with our herpes viruses, right? We secrete them in our saliva occasionally, not all the time. Some people secrete more than others. We find the same thing with the elephants, okay? The other thing that was interesting, though, in all these normal, healthy elephants, we didn't see any evidence of the virus floating around in their blood. So the fact that they were secreting some in their trunk secretions, but finding nothing in the blood means there's very low level. They were containing this virus. Nothing was too serious was going on with this virus. In contrast, though, and this is another simple graph. Let me just lay this out for you here. This here shows the amount of virus. This is actually a log scale, so this goes up to the millions here. And this is days here. And this intersection here, this is of a baby elephant that got sick from this elephant herpes virus. And these are samples that we got from an elephant that got sick back in 2009, okay? And the thing that I want to point out to you here is that this is when the baby elephant started to show signs of clinical illness here at day 0. There are two things important that this graph told us. First was you could see virus in the blood before the elephant had signs of getting sick, okay? And then when it did get sick, it had a lot of virus, almost like a million copies of this virus in just a very tiny drop of blood, okay? So lots of virus. But then this elephant, for reasons I won't get into right now, is it actually recovered. And you can see that it actually controlled the amount of virus that was in the blood, and ultimately it went down. And this elephant actually lived, okay? So what this told us was that for application to the Houston Zoo, we thought, well, what if we were to look at the elephants at the Houston Zoo on a regular basis? Could we find, if we found evidence of low levels of virus in the blood before they got sick, maybe we could start treating them before they got sick early, because once they get sick, most of the time it's too late, okay? So we ended up establishing then a regular monitoring program at the Houston Zoo, and now many other zoos throughout the country and in Asia's places in Europe and other zoological institutions now are using our tests to do the same thing, where they're now regularly monitoring the blood of particularly young baby elephants, susceptible young baby elephants, looking for evidence of this virus in the blood, low levels, so that they can jump on top of things and try to treat these elephants before things get to be too bad, okay? So we started doing this actually at the Houston Zoo, and I don't know if any of you saw, there was a newspaper article in the Houston Chronicle in October that talked about 2 cases at the Houston Zoo of Baylor and Tupelo, who you saw in the video earlier. Both eventually got infected with very serious infections with this elephant herpes virus. And it was through our monitoring program that this is evidence that our monitoring program actually worked. So let me go through again. These are similar graphs to what I showed you before. This is Tupelo, who got sick with EEHV in the fall. So what happened? We were regularly monitoring the blood. We didn't see anything going on. And then we saw a jump here of virus levels in her blood. And that signaled to the elephant keepers and the veterinarians at the Houston Zoo, we need to start treating this elephant as if it might be developing a serious EEHV infection, which in fact this elephant did. And you can see that levels started to climb. Treatment was instituted around about this particular area here, and you can see virus leveled off and started to go down. So this means the elephant started to control its infection to levels where ultimately after 2 or 3 months, this elephant had almost no evidence of the virus in its blood at all. Interestingly, you can look at the trunk secretions here. You can see that this amount of virus was shed, tremendous amounts of virus were shed in its trunk, which also were controlled by the animal after 2 or 3 months. We have some new parameters now that we also look at. You can look at other things in the blood. These are some cell counts. There are 2 different cell types that we look for in the blood. One is platelets, which is involved in clotting, blood clotting. And it's interesting, this is very characteristic of hemorrhagic disease. You can actually see when amounts of virus go up, the amounts of platelets actually start to drop. You can also see these types of cells, which we call immune cells or white blood cells, also drop. And this was coincided with the increase in the amount of virus that we see in the blood. Interestingly enough, when this elephant started to recover, these levels bounce back to higher levels, and then they go back to sort of normal levels. So we now have a pretty good picture through looking at blood cells and platelets, as well as monitoring virus in the blood of these baby elephants, where we can attack and where we can institute treatment of these baby elephants before they actually develop clinical signs. And we think that this particular process helped save Tupelo from her EEHV-associated illness back in the fall of 2014. I won't show you data, but her half-brother Baylor also had a similar instance of this disease within about a few weeks of each other. He had very similar types of data, and he also survived. So that's good news. Okay, so let me just summarize some of the things that we've talked about. What is EEHV? This is a newly recognized herpes virus that can cause sudden or rapid lethal hemorrhagic disease in elephants. Why should you care about EEHV? It's impeding self-sustaining population, lots of monetary and emotional cost. It actually costs about close to $60,000 per year per elephant. So if you have a 5-year-old elephant die from EEHV, that's a $300,000 investment. So just from money alone, it's a huge monetary cost. The emotional cost, though, is really devastating. Some of these elephant keepers who manage and take care of these elephants are with these elephants they spend more time with these elephants than they do with their family. So it's a devastating emotional cost. And this disease is affecting range countries and wild elephants in range countries as well. Why are we involved? Of course, who wouldn't want to save endangered baby elephants? What's our vision? We have this bench to barn program where we're developing diagnostics, treatments, long-term vaccine. We think that we're having some success with our diagnostic assays and treatment protocols to help save some of these baby elephants from this devastating disease, which is part of what we've done. And we've instituted weekly monitoring programs to look at these elephants in order to help monitor for this disease and treat them. We've also discovered that these viruses seem to be shed in their nasal secretions, and this may be the major way in which these viruses are probably transmitted between different elephants. Okay, so where are we going? What are we doing? I'd just like to talk about this in very general or broad terms. One of the things we've done, I don't know if you all are aware, but Baylor College of Medicine is one of 3 genome sequencing centers in the United States. And so they've got some very powerful technologies that we were able to take advantage of. But we were actually able to sequence the entire virus genome of the elephant herpes virus, okay? And so we're working on that. We've done that. And why are we doing that? Well, one of the reasons is we have to know what are the different components that are made by the virus in order to develop a vaccine. We have to know how the virus is made up and the different parts. In order to do that, we actually had to sequence the genome. So we've done that. And we now are actually studying immunology. Now, what is immunology? Immunology is actually the way that we respond to virus infections and control them. Many of you have heard, of course, of antibodies. Antibodies are a major way in which we help defend against virus infections or help control virus infections. There's another way that we help control virus infections. It's called cell immunity. So antibodies and cell-mediated immunity. And we're looking in quite detail about how elephants respond to this elephant herpes virus and what parts of the virus are most important for forming immune responses against that we could use to develop a vaccine. So those are the roads that we're going down right now. I'd like to also mention at this time, a lot of people ask me the question, does our research have any broad applications to human medicine or medicine of other kinds? And my first answer to that is that really, does it have to? I sometimes feel that isn't saving the elephants from this lethal disease a noble cause in and of itself that should be worked on? That's my first answer. So yes, but there are also some things that I think as a general principle, many people often forget, and that is science isn't necessarily a linear process. That we work on one thing is going to lead to all the great answers are going to come in a linear way. Lots of times important discoveries come from unexpected places. And I'll give you an example. So there's the PCR test that I mentioned that we use for detecting this elephant herpes virus utilizes a reagent or an enzyme. We call it Taq polymerase. It's not really important to know details about that. But the thing is, PCR, that test, is as important for people studying in medicine now as a screwdriver is for a carpenter. It's a critical type of thing that we do. Well, that enzyme that's needed for that, where did it come from? It actually came from people who are studying these bacteria that live in the thermal vents in Yellowstone. You know, the geysers that pop up? There's actually bacteria that live in those boiling cauldrons of boiling water that come out of the water. The enzyme we used for that test came from studying those bacteria. Who would have guessed that by studying that, we would have something that is absolutely critical for the types of things that we're doing to advance medicine in humans today? So it's maybe not quite as satisfying an answer, but I would say that studying the elephants and their unique physiology and their unique biology may have some unexpected types of things that may have spin-offs for other types of medical problems, okay? And I just leave you with this. My hope is that through our research program that we have baby elephants that can continue to keep doing this. This is actually at the Houston Zoo and one of the pools that they have. And there's Baylor acting like a typical big brother. And of course, we hope to have little elephants like Duncan here grow up to be as big as his daddy, shown here, Tai, who's actually the largest known Asian elephant outside of range countries in the world, actually. He tops out at over 13,000 pounds. So imagine that. He's about the equivalent of 2.5 SUVs put together. And before I finish, I just want to acknowledge some people that were involved that help us with all of this. Many people at Baylor College of Medicine, several people have come through my laboratory. Very talented postdoctoral fellows and graduate students have contributed to this. In particular, Jeffrey Stanton, the first veterinarian who came to work on this project. Other veterinarians, Sally Knopfs and Alan Humphreys. My current postdoctoral fellow, Angela Fury, who's doing some of the immunology work. We have some very important collaborators in the Human Genome Center, Johns Hopkins University, and in particular, the crew at the Houston Zoo, both the veterinarian crews as well as the elephant team who takes care of the elephants, some other zoological institutions, both within our country as well as around the world, and then also finally acknowledging some funding programs for which none of this would have happened. The International Elephant Foundation, the Houston Zoo, International Elephant Managers Association, a government organization called Institute of Museum and Library Services, and of course the Morris Animal Foundation. And with that, I'll take any questions. We generally define them as juveniles or babies up to about the age of about 9 or 10 years of age. That's generally the age when they reach sexual maturity. So females can be bred between about 8 and 12 years of age, and males are capable of breeding at about that time frame. Most of the susceptible baby elephants that are susceptible to this disease, though, are between the ages of 2 and 7. Now, you might say, why not younger than 2? And we believe there may be some component of maternal immunity that's transferred from milk. And so they generally wean from their mothers at about 2 years of age. So we think there might be some component with that helps protect the baby elephants until the time they reach about 2 years of age. So that's actually a really good question. So the question was, can the virus be transmitted to humans? Not that we know of. That's actually a really good question. And we're developing tests to be able to look at whether or not an elephant has been previously infected or not by being able to look at their antibodies, for example. So if an elephant has antibodies, detectable antibodies to EHV, we know it's been infected before. And so we could actually apply that test to look at people. I can tell you that that's actually something that a lot of elephant keepers are like, well, I'm not sure I want to know the answer to that. On the other hand, so far, we have not had any evidence that any known disease can be caused by this virus in people. The other thing to remember with herpes viruses, I didn't tell you about before, they're actually very specific for each species. They generally don't cross. Like generally, a herpes virus in zebras would have a hard time infecting people. They've evolved to actually be very specific for the host that they infect. They can cross species, and initially that was actually thought maybe to be what was going on here with the elephants, because it actually historically, a lot of zoos actually used to keep Asian and African elephants together. And one of the thoughts was that it was an African elephant virus that jumped over to Asian elephants, and that's why it caused such bad disease, because it wasn't naturally found in those elephants. It turns out that's actually not true. So most of the time, these viruses stay most of the time within the host that they're found in. So it's a good question. Yeah, that's a really, really good question. So the question was, have any of the elephants that we've detected that got this disease relapsed? The answer so far is no, that if they get infected with this virus, they're protected from this virus thereafter. There are different, I didn't tell you about this, there are some different flavors of EEHVs. We call them 1, 2, 3, 4, 5, and 6. It does look like if an elephant gets infected and sick with one of them, it's not always protective for them getting infected and getting sick from another one of the other different flavors. So we don't quite know the answers to that, but that's a great question. Yeah. Yes? Yeah, so these are all really, really good questions. So the question was, is there any gender-specific difference between susceptibility to getting infected or getting sick from the EEHV virus? And so far, it seems to strike universally. It doesn't really matter male or female. The second question was, is the frequency by which this is happening in captive herds different from what we see in the wild? And the answer is we don't know, but I will say this. Because of habitat loss in the wild, many of the herds are becoming much more fragmented, so they have smaller places to live in, and they're becoming more isolated. So smaller herds that are more isolated. So the herds that are existing in the wild today look actually a lot more like the smaller herds that we are tending to keep in captivity. So I suspect that one of the reasons why we may be seeing more of this going on in the wild potentially is because of this particular issue. So, and it's also important to remember just to try to distinguish between wild and captive elephants. In Thailand, for example, you might think, oh yeah, there's wild elephants running around the woods all over the place. The fact is, more than half of the elephants in Thailand are under the care of humans in one way, shape, or form.